Hello everyone, today's video is for ExCultBaby, so let's jump right in. Photosynthesis is a cellular process by which sunlight is used to stimulate the production of organic molecules that will be utilized for cellular energy. There are two kinds of photosynthesis, anoxygenic and oxygenic. Microbes that perform anoxygenic photosynthesis, such as green sulfur bacteria, purple bacteria, and acidobacteria, use hydrogen sulfide as their final electron acceptor and produce elemental sulfur. Microbes that perform oxygenic photosynthesis, evidently only the cyanobacteria, and, by extension, their endosymbiotic partners, use water as their final electron acceptor and produce oxygen. The origin of oxygenic photosynthesis had massive consequences for life on Earth. Of course, the evolution of oxygenic photosynthesis wasn't an overnight development, and as it is a cellular process, the odds that its evolution would leave behind fossils is pretty much non-existent. However, there is an interesting twist. The evolution of oxygenic photosynthesis did leave behind something, well, some things, in the geologic record: oxidized manganese and banded iron. Why is this important? Prior to about 2.1 billion years ago, there was virtually no free oxygen in the atmosphere or oceans. However, a deposit in South Africa, dating to about 2.4 billion years ago, contains oxidized manganese. Paradoxically, this deposit also contains sedimentary pyrite and uraninite, indicating that no free oxygen was present. To quote the book A New History of Life by Ward and Kirschvink, quote, "So what could the resolution of this paradox be? We think that the oxygen-emitting system of cyanobacteria had not evolved at this time, 2.4 billion years ago, but that many of the evolutionary steps needed to get there had already been taken. It turns out that the actual biochemical complex in oxygen-releasing photosynthesis that collects the energy to split water, releasing oxygen, relies on a cluster of 4 manganese atoms, with a calcium atom thrown in for good luck. When this protein is made from scratch in living plants, the manganese atoms are sucked into the complex, one at a time, with the aid of photons that oxidize them. We suggested that these unique bursts of manganese in the sediment, not timid whiffs, might be the product of an evolutionary ancestor of the cyanobacteria that fed on reduced manganese dissolved in the water, using it as a source of the electrons needed to do photosynthesis. Many primitive photosynthetic bacteria are known to do this with hydrogen sulfide, organic carbon, and ferrous iron, but none have yet been found that can use manganese. Photosynthesis of this sort would leave copious amounts of a waste product, manganese oxide, behind in the sediment, but would not release the molecular dioxide that would destroy the sedimentary pyrite or uraninite, or create an ozone screen to change sulfur chemistry." Close quote. Once photosynthesis did evolve, cyanobacteria began pumping free oxygen into the oceans, which caused the rusting of iron by about 2.1 billion years ago. Once the available iron was exhausted, free oxygen could even outgas to the atmosphere. All this reflects interesting things going on in the biome, including evidence for early eukaryotes. The oldest probable eukaryote fossils, known as acritarchs, date to 1.8 billion years ago, and this approximately accords with the date of origin for the domain Eukarya. The domain itself is broken into essentially 4 supergroups: the bizarre excavata, including Euglena and Trypanosoma; Archaeplastida, the plants and their algal kin; Chromista, including forams, diatoms, and kelp; and Amorphia, including amoebozoans, fungi, and animals. Some of these supergroups may not be proper clades, and the relationships between them are still being worked out by researchers. The origin of Eukarya has been hypothesized to have occurred as a result of a methanogenic archaean forming a symbiotic relationship with an alpha proteobacterium. This bacterium became enslaved to produce cellular energy, large quantities of the molecule adenosine triphosphate. eventually allowing eukaryotes to increase both their genome and cell size. Later, a second endosymbiotic event occurred in which a lineage of eukaryotes took in another bacterium, this time a cyanobacterium, to do oxygenic photosynthesis. This lineage would give rise to red and green algae, as well as ultimately land plants. But endosymbiosis didn't stop there. There are eukaryotes that entered an endosymbiotic relationship with cyanobacteria independently, one being Pollinella chromatophora. Other eukaryotes like Euglena and chlorarachniophytes obtain the cyanobacterial plastids indirectly by entering secondary endosymbiotic relationships with green algae. Eukaryotes of the aforementioned supergroup Chromista underwent secondary endosymbiosis with unicellular red algae. These chromists include photosynthetic organisms such as brown algae like kelp, diatoms, coccolithophores, and dinoflagellates. There are also chromists with red algae-derived plastids, but these lost the ability to perform photosynthesis. These include the Apicomplexa group. One of its members is the infamous parasite Plasmodium that causes malaria. Other chromists took endosymbiosis even further. One species, Rhopalodia gibba, replaced its endosymbiont with a cyanobacterium. A few dinoflagellates in particular replaced their plastids with green algae, or even engulfed other chromists, entering a tertiary endosymbiotic relationship. One interesting line of evidence for this relationship is that some protist host cells possess a vestigial nucleus called a nucleomorph, which once belonged to the endosymbiont in addition to the host's nucleus. While we are certain that for some eukaryotes the endosymbionts were obtained by engulfing other eukaryotes, exactly how isn't certain. This is especially the case for the chromists. As mentioned, chromists contain non-photosynthetic groups that still have their plastids, but there are also chromists that don't show any sign of ever having possessed plastids at all. The question is whether there was a single chromist ancestor that engulfed a red alga, and later multiple lineages lost their plastids, or whether different chromist lineages engulfed a red alga independently. Some researchers have proposed the Rotoplex Hypothesis, where the red algae plastid was passed on from one chromist lineage to another, and so on, in a series of endosymbiotic events. There are likely other undiscovered protists who too have developed endosymbiosis with algae, and possibly many others throughout geologic history, some with lineages lasting 100 million years, that we will never discover because they did not have the good fortune to be preserved for posterity. While no animal possesses endosymbiotic plastids, many do have symbiotic relationships with protists and algae. As we discussed in the video Ocean Acidification, corals house dinoflagellates. That means a coral contains a dinoflagellate, which contains a red algae, which contains chloroplasts and mitochondria, themselves the descendants of bacteria. It's basically a living Matryoshka doll with smaller organisms providing nutrients up the chain and larger organisms providing housing down the chain. The acoelomate flatworm, Sagitifera roscoffensis, also utilizes the green algae Tetraselmis convolute for energy. See the Creation Myths video linked in the description for further discussion of this subject. Other organisms have recently been shown to be cocktails of symbioses, such as lichens. Typically, lichens are regarded as a symbiosis between at least one fungus and at least one photosynthesizer, whether algae or cyanobacteria. However, recent research shows that lichens possess specific species of bacteria that may act as a biofilm, and more than a single fungus may be present in the lichen. For example, a 2016 paper showed that while the Ascomycete bryoria and green algae Trabuxia form the body of the lichen, the yeast, Scyphobasidium, takes up residence inside the body, surprising mycologists. Humans, too, house various bacteria and archaea, especially in the gastrointestinal tract, including members of the phyla Bacteroidetes, Firmicutes, Actinobacteria, Proteobacteria, and Verrucomicrobia. These can break down foods into certain metabolites, and those metabolites can have pretty major effects on our health. For instance, researchers shown that dietary fiber is broken down by our gut microbiota into short-chain fatty acids and succinate, which help regulate gut homeostasis, thus preventing obesity, non-alcoholic fatty liver disease, and type 2 diabetes mellitus. But let's get back to the point of the video. Clearly, endosymbiotic relationships exist throughout nature, all eukaryotes have at least one. Many organisms have endosymbiosis with chloroplasts, but some organisms steal chloroplasts from others. This is known as Kleptoplasty, and it's been identified in a few organisms: sacoglossan sea slugs, marine flatworms, and at least one dinoflagellate. In essence, the kleptoplast consumes an algae, but rather than break the whole thing down, the kleptoplast retains the chloroplasts for a period of time, using them for energy. This has led to what are basically photosynthetic animals, such as the sea slug, Elysia chlorotica, which feeds on the green algae Vaucheria litoria. Sea slugs are evidently able to maintain their stolen chloroplasts anywhere from 57 days to an entire year, and researchers have wondered how this is possible. At present, there are hints, but no definitive answers. Some researchers hypothesize that horizontal gene transfer from the algae to the slugs had occurred, allowing the latter to possess the chloroplasts for long periods. However, this appears to be largely false. Most kleptoplastic slugs have no algae-specific genes. The few who do don't seem to have any genes for maintaining chloroplasts. Instead, as a 2019 paper explains, quote, "Long-term plastid retention is now thought to be the result of the robustness of the plastids themselves, the stability of their essential proteins, and the physiological adaptations of the host." Close quote. Clearly, there is still much to learn about these animals and their adaptations. As for marine flatworms, the species Bicolellia celaris and Pogena paranagulgus steal their chloroplasts from diatoms, and in the Arctic, a species of dinoflagellate has been observed stealing chloroplasts from haptophyte algae. Kleptoplasty outside of sacoglossan slugs has only been known for a few years, so one must wonder how many more undiscovered kleptoplasts are out there. In a sense, kleptoplasts are a step towards endosymbiosis. They are dependent on chloroplasts, at least to an extent, and can retain the chloroplasts for a substantial amount of time. How long until those chloroplasts can reproduce inside animal tissues and be passed down through successive generations? Time will tell. As we can see, chloroplasts are very useful organelles that are utilized by various lineages of organisms, where the whole, indeed, becomes greater than the sum of its parts. So, thanks for watching, and I'll see you next time.